All right, let's go ahead and talk about the pathogens now. And so, kind of from quiz 2.1, we already kind of went over these, right? Just a little bit. We got bacteria, we've got fungi, viruses, and nematodes. And so these particular pathogens are going to attack mostly either the roots because they're kind of soft, they possibly the shoots because that is where there's a lot of nutrients, and then also the stems because there's woody material there. And they're going to attack our field crops. Fruits and vegetables, especially, are susceptible to these pathogens because most of them are soil borne. And if you think about it, some of our fruits and vegetables, at least the vegetables anyhow, something like squash, fruits being watermelon, any of these vegetables that are really close to the ground are susceptible to these pathogens. And then also our turf grass is especially susceptible because they're so low to the ground, and with these pathogens being soil borne. So we're going to start off with bacteria. If you remember from soils, we talked a little bit about some of these different uh, soil microbial populations. They have some benefit to our soil health, but they can also cause some very serious damage to our crops as far as economic impact. And so they're designed in order to break down some of these organisms, these plant materials, and release those nutrients back into uh, the soil and the environment. They're typically single cell, so because they're single cell, they're going to divide, and that's how they multiply. Um, they don't have a true nuclei compared to animals or plants, and so this can impact how that DNA molecule is suspended, how we're going to attack uh, how we're going to combat this, right? So there are several different bactericides, antibiotics that we use that are specifically designed to attack these bacteria. Not so much about the shapes, but understand that the shapes are important because what happens is there are receptors inside of the cells, and these bacteria kind of insert themselves into those shapes. So think about square peg, round hole. If it's a square peg, it's not going to fit in the round hole. But if it's a spherical, like a coccus, like a spherical, then it is going to insert itself in those places. So just a look at the different shapes. We've got rods. We've got a rod on a root hair, and this is important because um, if you look under the microscope and you can identify the shape, it kind of gives you a diagnostic tool in order to determine which one of those different species is actually attacking your plant. And once you know the species, then you can go to the proper bacteria side in order to combat this. So most of them are going to be aerobic. And if you remember from soils we talked about, we've got obligate and we've got facultative. And so obligate require either aerobic or anaerobic conditions, whereas facultative can survive in either or. They have a preference, but they can survive in either or. And so if you know that it's an aerobic, or let's say it's an aerobic bacteria, that you know that it is, how do you think that you would go about just in general, what would be the first thing you would take away from this particular bacteria? Yeah, oxygen. If it's facultative though, and it can grow with or without, then you might need to do something different in order to eradicate that. So, another important factor for identification is whether it's gram positive or gram negative. And so, if you were to have an issue, um, you would send that off to the plant diagnostic lab. The first thing that they would do is they would look at it, determine if it's likely to be a bacteria or a fungi or a virus or whatever, and then they'll look under the microscope, and then they'll test for gram positive or gram negative, and that gives them a diagnostic tool in order to determine what the next method of um, what the next step in the process is. So we have signs and symptoms, and so the sign is something visual that the symptom is something visual that you'll see, and then the sign is actually the when you look at that under the microscope. So one of the first things that we'll see are these bacterial galls, or maybe even some of the ooze. And so what happens is that bacteria infects that cell. They break down the cell wall, and what that does is everything just kind of leaks out and is just kind of free flowing. And I mean, you got to think about the inside of a cell and the cytoplasm being sort of this kind of like gelatinous, oozy sort of material. And once that cell wall disrupts, then now that is just free flowing throughout the plant, and there's no structure. So you might see these water soaked lesions as well. What's important is to understand the environmental conditions. And so, in general, most of our bacterias and our fungi um, are going to prefer warm, moist conditions because that's their ideal environment. And so, the way that that gets in there is, we talked about this in 2.1 is, through the stomata because that's an opening. We've got the lenticles of the hydrathodes. And when we have windblown soil, especially sandy soils, that's going to cause these little bitty breaks. Like imagine, like, on your hand, if you have these little openings, that's an opportunity to get an infection. So making sure that we keep our wind erosion to a minimum is going to help us overcome or at least minimize the potential for us to have some bacteria infection. As I mentioned, these enzymes are going to degrade that cell wall, and so then they're going to infect the tissues. These enzymes, like I said, they're going to degrade that cell wall. You're going to see that bacterial ooze, and once you see that ooze, then that kind of gives you an idea that you have now a bacteria infection. So those complex sugars are going to actually plug the conducting tissue. And so if you remember, if you had me for plant science, we talked about that the xylem is important for taking nutrients through the soil, out of the soil, in the soil water, up the xylem to the leaf to perform photosynthesis. And so photosynthesis makes carbohydrates. The carbohydrates then travel throughout the plant via the phloem. And so if you plug those conducting tissues, then the plant doesn't get the necessary water, nutrients, and carbohydrates that it needs. All right, so there are different types and different ways that that infects. And so if you had me for 4120, we talked about that we are using agrobacterium and our TI plasmid to infect that cell. And once that cell is infected, that the genetic modification occurs inside of that plant cell and then is transferred throughout that plant. We have bacterial wilt, where we have now just degraded this cell wall. They're just gone. And so, if there's no connectivity between these cells, then we start to have voids. And then finally, bacterial wilt. So that, uh, as I mentioned, that we have water moving up the xylem into the plant, trying to get to the leaf. And so if we have these bacteria that have either degraded the cells, they're not able to perform their function, or if they are causing a blockage. And so think about this much like an artery for a human. If we have a blockage, then the heart can't pump the blood to the necessary parts of our body. If there's a blockage in the xylem or the phloem, we can't get water or nutrients spread throughout the plant. Just a better look at that. This bacterial wilt is going to be a clear sign that you have like that wilting and being and watering even if the soil is moist. Again, because it's blocked, we cannot get water and nutrients to the appropriate parts of that plant. There are about 200 species of these bacteria, and they're going to be either in the plant, on the plant, in the debris. They're going to be on dead stuff. They're going to be in soil. And you have to think about soil being vast wide, like an area. Think about having bacteria on an acre and think about how deep that bacteria could go. And then how do you combat that typically when you have a bactericide that's in soil, you're going to need to drench that soil. So think about how much water you would require in order to apply this bactericide. One of the problems with bacteria is because they're single-celled organisms and they're growing, they're going to reproduce at a very rapid rate. And so you can have, each species can, or some species can divide 20 minutes, every 20 minutes. So you're getting almost an eight, what is it? So from, let's say we had 100 at minute one, and then we have 200 at minute 20, and then 400 at minute 40, and then 800 at minute 60. So we're getting, what, an 8x increase in um, division over an hour. So we could, now let's scale that up because they're very small, microscopic, in that you can get almost a million, like produce another million of these in 24 hours. And so in 24 hours, we get one. In 1 week, we've now got a 7x, 7 million. And then in a month, you've got now 28 million of these bacterium. So if you get a bacteria infestation, you really kind of need to get at it very quickly. Unfortunately, if you think back to the blockage part a couple of slides ago, once you have that blockage, it's very difficult to get that into the plant and then also overcome and kill those bacteria. All right, so they're going to survive on inanimate objects in water. So that's why, like, water movement, water drainage, and then also wind flow is important for all of these different diseases. So they can be disseminated through the air, through insects. We talked about it earlier. We talked about insects as not only attacking the plant, but also carrying different pathogens as a vector. Through water and soil. And then also humans and other animals can carry these bacteria as well, particularly in like greenhouse situations, in that if you cut a plant that has a bacterial infection with a pair of snips, and then you go to cut another plant with those same snips that are infected, you've now basically inoculated that new plant, and you're continuing to cut and you're continuing to use those. And so if you're in a greenhouse environment, it's important to disinfect your equipment. So some of the signs and symptoms of a bacterial infection are going to be very similar to like a fungal plant. So it's important, to understand that a symptom is something that says there's a problem. It's something that stands out that goes, wait a minute, that's not right. The sign is an actual physical evidence of the problem. And so you're going to see the symptom, and then you're going to need to address the sign. So we're going to have leaf spots and blights, cankers. These are obvious. We've got scabs. That's going to be another symptom that you can go, you know what, this is likely a bacterial disease and not a fungal disease. And then the wilt as well. So one of the most common symptoms is going to be a leaf spot. You're going to notice this clearly. It's just not going to be green. It's going to look off, and that's just not something that you would want to have. Spots on your leaves. Like, there's an issue here. What is the issue? And if you get into a career where you're going to do that, like as far as like plant pathology or maybe entomology, the more that you go through this, the better you'll become at that. So my goal for this course is to get you introduced to the different components of plant pathology and crop pests and disease. And if you make it into that field, you'll gain the experience the more that you do that. You're often going to have this like sort of rotten or fishy sort of smell. Like you'll be able to smell something is wrong because everything's going to be sort of like disintegrating. Like think about Think about like having like a bucket of soil and just water logged and it's just rotting and nasty and just bleh. And so that's going to be some symptoms that you'll recognize. You'll see that bacterial ooze. That's going to be one of the telltale signs that you have a bacterial issue. Is that ooze? You'll have this chlorotic halo. So there will be, and we'll see this in a couple of slides later. You'll start to see that around those spots. And so those spots will start to coalesce and they'll move towards each other. And that's going to cause large areas of necrotic tissue. And so think about it. Like once that happens, that tissue's not going to grow back. And like think about just having a big hole in your leaf. Like how does that leaf repair that? So pretty much once you get this bacterial issue, this bacterial disease, like you're really stuck. And so what you want to do is minimize the spread of that so that if you did replant or that it doesn't spread, and you're kind of stuck. Most of the bacterial, almost all of the bacterial leaf spots and blights are caused by two species. And because we know those species, Pseudomonas or Xanthomonas, we know what their background is. We know what their shape is. We know if they're gram positive or gram negative. And then we have chemicals, we have antibiotics that we can use in order to combat that. So for cankers, we're going to see that on the trunks and the stems. They're going to have that sunken dark brown. We'll see this very clearly in the next slide. And it might be that that canker is like orange or brown or very dark brown. It's rotting, right? So many of us have seen this. This is kind of in the armpit of that tree. And we start to have this sort of degradation of that. And you can see this ooze and this nastiness. It's just like, ugh, you don't want that on your plant. And you don't want to get that on you. So think about it like that. Like you wouldn't want this on your skin and you wouldn't want to transfer that. We'll see these lesions and just all the cells in this little area have just disintegrated. They're just, how do you repair that? How do you overcome this? It's probably not going to come out of that. And so you just want to minimize the spread into the next growing cycle. So we talked about that gum exudation. That's just, we're going to find that on the twigs and the branches. You'll find galls as well. They'll be soft. You wouldn't want to touch this, right? Like you're not just going to, you wouldn't want to walk up and poke this because you know that you're going to get that gunk on your hands. All right. And so moving into bacterial galls, we've got agrobacterium. We talked about this earlier in that agrobacterium is going to infect that cell and it is going to impart some of that DNA into it. So that agrobacterium can be beneficial, but in 4120 in crop improvement, that's how you know that the agrobacterium is doing its job is you'll see that gall. We have Rhizobacter. We've got Pseudomonas. These are prevalent throughout soil. The Rhizobacter are also important for nodulation. So we have these different types and different species of these bacteria, some beneficial, some not so beneficial, that are causing issues in our plants. So these galls, you might hear them as crown galls, as crown knot, as root knot, root galls. As we move into nematodes, you'll see these as well, because they're just going to be these knots. That's going to be the telltale sign that you've got a bacteria infection. There needs to be a wound in order for that bacteria to enter. Uh, um, you know, Windblown soil that causes a little nick in the leaf, or like maybe in nurseries, we're coming by and we're cutting, that opens up a wound. And so that is how that bacteria gets into that tissue. Remember that we can have rapid and continuous cell division and that very quickly that bacteria is going to multiply and spread and degrade that cell wall. So that crown gall is going to be small, white, soft, and round. And you'll likely see that on the main root, not so much on the root hairs. If you do have this bacterial infection, you probably don't have root hairs because it's degraded all that cell material, and those cells can't divide. That cell division and the zones of elongation and just the creation of new cell tissue. All right, so here's a look at our bacterial galls. We saw this one earlier. Here are some galls on our roots. And so you can see that there are not a lot of root hairs on this. These are the main root tissues, but that brown, that sort of golden, that's not what they should look like. You know they should probably be more white and soft, but these are not. These are hardened. Notice no root hairs. We've got these galls. So it's possible that you see some issue above ground. If you have a symptom above ground and you don't see that any sign above the ground, then you'll likely need to dig that plant up and see if there is a root issue. Next, we have vascular wilts. And so this goes back to originally when we talked about that if you have this blockage of those vascular systems, the xylem and the phloem, then you're going to have some blockage and you can't get enough nutrients and water. If you don't get enough nutrients and water, you don't perform photosynthesis. No photosynthesis, no nutrients, no new plant growth. Here we are with that translocation of the nutrients and the water because those cells are now gummed up. And so they'll destroy that cell wall, the xylem vessels, some cellular debris. There's just junk in those vascular systems. And once that junk's there, you're pretty much done because once that plant reaches wilt, it's very difficult to get it to come back out of that. So wilting and death of the above ground part could likely, think about it. Where would you expect this to stem from? Probably from the roots because the roots are what's taking up the water. And if there's bacteria that there's soil bacteria that is in the water that make it into the plant, you now become, compromised from the inside out. We'll get that bacterial ooze that'll come out of the stomata. You'll see that on the leaves. And once you see that ooze, it's pretty much over with because that plant tissue is dead. It's like the plant trying to expel all of that nastiness to get it out of itself. But that tissue's already dead and it's already lights out. Try to minimize the spread of that for the next growing season. So here we have a look at what a symptom would look like. We've got the symptom here and that our plants are just wilted. Something's wrong. We've applied water and nothing has changed. It has not come out of this. That's a telltale symptom that you have a bacterial disease. You dig up your plant, you see that your roots are, just sloughed off. There's not a lot of strong root growth. You open up the plant and you see that the inside of that, you now have basically rotted out your xylem, and you can start to see that the phloem is also being rotted as well. So this is just not going to translocate nutrients and water very well. Next, we have soft rots. And in the next couple slides, you'll see that. And so one thing about these soft rots is that these bacteria are prevalent almost always, in that it just takes time in order for the symptoms to occur. And once the symptoms occur, you likely cannot get back out of that. So, this soft rot is not necessarily a phytopathogenic, meaning a plant pathogenic bacteria, but that it is more of a saprophyte in that it's going to try to decompose whatever tissue is there. So those soft rots have, across a lot of different hosts, they cause disease in those fleshy plant structures, both above and below ground. And because we know like what species cause these rots, we can then use the proper maybe antibiotics or bactericides in order to combat that. So when plants are under stress or even being stored, and I believe in the next slide, the next couple of slides, we're going to show where, like when you see the next couple of slides with the pictures, you'll see that when they're being stored, they're susceptible to these, just basically rotting. And so we'll have that bacterial ooze. They enter through the host wounds just like anything else. Those cells, those cells disintegrate. They become watery and soft, and you'll have this sort of slimy, foul-smelling ooze that is causing that. Like that's the symptom. All right, here we go. Here's our bacterial soft rot. If you've ever put, a bag of potatoes in a cupboard and you just decided that, you were going to go on keto or something and you're not going to eat potatoes anymore. And then let's say you're going to clean that out. Maybe this has happened to me. Okay. How about that? I've put a bag of potatoes in a cupboard to keep them away from the light so that they don't grow those little sprouts on the eyes. But what happens is those dark, sort of warm conditions are suitable for these bacterial soft rots. And so the fruit just gets mushy or the vegetable just gets mushy, right? And so this happens over time. And if you've ever left a vegetable for too long, then eventually that bacteria that's already there will start to divide and it will rot that fruit or that vegetable. And so, these can be an issue. Uh, these can be an issue in that if fruit or vegetables sit too long, that they'll end up spreading it to other fruits and vegetables. And so we want to make sure that we don't have this because this is not sellable. Some of the control methods for our bacteria is going to be very challenging in that once that bacterial disease occurs, it's very difficult to come back out of it. And also remember that those bacteria spread in millions per day. Like up to a million different, like enough, up to a million increase in population from 1 to 2 million in 24 hours. So what we want to do is look for plants that have like genetic host resistance. We can build this in so that our cell walls are stronger to attack against that pathogen. We have cultural practices, chemical application of biological controls, which we'll talk more about in module 3. So we have genetic resistance. We can see that it's already been genetically modified, and we can see that it is resistant to these different bacteria. Here's a look at our tomato plant that's been genetically modified or has some resistance to that, and this plant does not. So we would want to move this plant forward and make sure that we don't get this one crossed in with anything else. You've got to make sure that you sanitize your pruning tools because you don't know if you have it or not. That's the thing, is you don't know that you have a bacterial disease until it's too late. We want to use crop rotation, reduced overwatering, because that's how that bacteria makes it into the plant. Minimize those surface wounds and make sure that you've got bacteria-free nursery stock. Chemical applications, we've got, a Bordeaux mixture, which is copper sulfate and lime. We can use antibiotics. Sometimes this is more of a preventative in that we're going to spray this to make sure that we don't have that bacterial disease. We can use antibiotics, but that's going to cause an issue because we know how people feel about just society in general about having these antibiotics in our foods. Because if we have the antibiotics in our foods, then we start to develop a resistance to the antibiotics. And if we get an infection, those antibiotics won't work anymore. All right, so that's going to end it for bacterial diseases. Remember, we've got leaf spots and cankers. We've got our symptoms on we've got our symptoms. This is something that stands out that says there's a problem. And then we need to look closely and see where is the sign? What is the sign of this? So the symptom is going to be something like this blackness, and we see this. The sign is going to be actually sending it off to the plant lab and having them determine what actual species that is. And then we have our soft rots.